Hello, and welcome to our presentation on autoclave validation. The purpose of this project was to find a new method to record temperature within the autoclave in the biochemical engineering lab and perform a validation. The previous method of recording temperature included a traditional wired thermocouple that caused an incomplete seal, invalidating the results. Sterilization is the process of making something free of bacteria or other living organisms. Autoclaves are used to sterilize media and waste to ensure that there is nothing present before an experiment or disposal. The issue of sterility caused FDA officials to put forth the concept of validation in 1979. Equipment validation documents are evidence that it conforms with the required standard of good manufacturing practices. It guarantees that the equipment consistently provides products of necessary quality. FDA Regulation 21 CFR 211.182 details equipment cleaning, use logs, and validation requirements. USP 1211 describes the basic principles for validation and certification of sterilizing processes. ISO 17665 specifies requirements for development, validation, and routine control of a moist heat sterilization process for medical devices.
Autoclaves are some of the most common types of sterilizers. This is mostly because steam is cheap, safe, and easy to produce. Steam is also able to destroy all types of microbial life because the steam denatures and coagulates proteins. This is useful for killing bacteria, endospores, viruses, and destroying prions. Steam is also able to penetrate all kinds of surfaces and materials, allowing it to sterilize multiple layers of cotton. Solid parts that are too sensitive to be sterilized in high-temperature ovens are also autoclaved. Denaturing and coagulating proteins require saturated steam. Liquid water and superheated steam are not effective at denaturing proteins, leading to reduced lethality when saturated steam is not present. This is caused by liquid collecting in dead legs, low spots in pipe runs, and condensate flowing down a pipe without insulation. Superheated steam can also occur if there is unexpected heating of the stream or if there is an unaccounted for pressure drop in the system. These issues can all be negated by eliminating dead legs in autoclave design, angling pipes and systems, and adding insulation to steam piping. Lethality is greatly dependent on temperature, as seen in the diagram on the bottom right hand of the screen. Temperature can have a logarithmic effect on how effective steam can destroy organisms. Validation is a critical technique that keeps everyone safe. Validation helps companies to ensure that the products they produce for patients are safe, effective, and produced to the proper industry and safety standards. Validation is a must-have procedure in pharmaceuticals, but is also extremely critical in the food industry. Other industries also use validation to ensure that their equipment works properly before introducing it into the production line.
Autoclave tape is a chemical indicator that changes color when exposed to heat, typically 121 degrees Celsius. Most autoclave tapes start a white or beige color and show dark brown or black diagonal stripes after a completed cycle. An example is shown on the slide. Our group chose to purchase the high-temp 140-1 temperature recorder. A picture of the device can be seen on the slide. To validate the autoclave, we ran a normal cycle with a waste bag and temperature recorder. To achieve validation, the autoclave must run at 121 degrees C for at least 30 minutes. The autoclave in the biochemical engineering lab achieved this metric, to be discussed more later in the presentation.
Kill curves are a way of figuring out how long the sterilization process will take at a given temperature to kill all the microbes or bacteria present. Or it can be used to figure out what temperature is required to perform the sterilization process in a certain amount of time. These are theoretical calculations, usually shown in the form of a graph. The standard temperature for autoclaves is 121 degrees Celsius or 250 degrees Fahrenheit, though the temperature could also be higher for shorter sterilization times. The graph shown here is just an example of what one of these curves could look like. The y-axis is on a logarithmic scale, meaning that each horizontal line is a factor of 10, or 1 decimal place, off of the next closest horizontal line. The starting value for the number of microbes or bacteria present can either be an assumption or a calculated value. The letters A, B, and C on the graph represent different temperatures that the process may be run at. As you can see, different temperatures take different amounts of time to theoretically kill all the bacteria or microbes. As a side note, because we are using steam for the sterilization process, these kill curves are referred to as thermal death curves.
One of the ways that we measure sterility is through temperature. and we measure temperature with thermocouples. And a thermocouple is an electrical device consisting of two dissimilar metals. And dissimilar conducting metals naturally generate a voltage that's dependent on temperature. This is known as the Seebeck or the thermoelectric effect. This voltage that they generate is directly proportional to the temperature of the system. As you can see from the equation on the left, the voltage of the system is equal to your Seebeck coefficient times the temperature of your system. So if you know the voltage and you know the Seebeck coefficient, then you know the temperature of your system, which you're trying to measure. And thermocouples are just metal wires, so they're very inexpensive and interchangeable and can be used in just about any system. Different types have different accuracy, and they all go down to about 1 degree Celsius of accuracy. On the right, figure 1 is our thermocouple that we used for our autoclave validation because it doesn't break the autoclave seal and sits as one unit inside the autoclave, whereas figures 2 and 3 are some examples of other types that you might find in the field. While thermocouples are a very quick, easy, and cheap way to measure temperature, they do have some issues associated with them. Circuit construction can be an issue where having an error in your reference temperature is going to adjust what temperature reading you actually see. Metallurgical grade can be a problem because Seebeck coefficients are very specific to metal composition, so if you have any factory errors in the metal, that'll affect the temperature reading that it gives. Grounding can be a problem as well because thermocouples can only be grounded in one place, and grounding in more than one can actually generate an electromagnetic field. And then aging is a problem as well because any metal that's going to be exposed to elevated temperatures will have compositional changes, and we see this in a Seebeck coefficient change, and thus again changes your temperature reading.
Because our autoclave is critical to the function of the biology labs in Faywick, it is important to validate the autoclave. As an example, a normal waste cycle was operated, and the temperature was recorded by a thermocouple inserted into the autoclave. Along with the autoclave tape on the waste as a validation test, the autoclave was able to hold a temperature of 121 degrees C for at least 30 minutes according to the temperature recorder. This shows that the autoclave is able to hold the required temperature for sterilization, and the device can be used without maintenance. On the graph, the accumulated lethality is displayed. This is an equivalency value that can be used to calculate the death of organisms as discussed earlier. The thermal death curve appears on the bottom right corner of the screen. If A, B, and C represent different types of organisms instead of different temperatures, then our autoclave would theoretically kill all of organisms A and B, but not organism C. With regular use, we also know that the autoclave is not getting too hot, running too long, or running too cold, because the sugars in the media made in the lab have not caramelized and the media has never had non-sterile growth.